Hi guys, I'm Akash Raut and welcome to Akash Biology. In this video tutorial, we're dealing about what gastrulations. That is the continuation of our previous video that was on blastula and its types and its fate maps. So we have to know what actually is what gastrula. So gastrula is what the movement and rearrangement of the presumptive organ forming cells that was formed in the blastula. This means in the blastula we have just made out of fate or that the animal pole will give rise to what the ectoderm. Among the ectoderm, the upper part will give what the epidermis and the lower part will give what the neural ectoderm. Similarly, the gray crescent will give the mesoderm, that is the notochord and the precaudal plate, and tail mesoderm, whereas the lower part, that is the vegetal pole, will give the endoderm. That was our assumption in the what the blastula. But how this process is being taking place, how the animal pole will form ectoderm, how the gray crescent will form mesoderm, and how the what vegetal pole will form endoderm, this process, all of this process is summed up into the what gastrula. So it is a what self-determined interdependent process and often known as what morphogenetic movement of cells. What morphogenetic movements of cells, that is, it will create a new relationship with among each other of the various germ layers. So in the gastrula, what actually is formed? Formation of three germ layer. What? Formation of three germ layers takes place over here. So it is a morphogenetic movements of cells. As we have discussed what it is a morphogenetic movement of cells, so the movement can occur in various types. What? Various types. So there are mainly what 5 types of movements that occurs in what the gastrulation process. So first of all, that is what invagination. What? Invagination. So what actually is invagination? Invagination is a inward folding or the infolding of cells to form a new cavity, and that the new cavity is being lined by what the already folded cells. So what actually is what invagination? Let me draw the figure. So there are the various cells overlying. So let's say this is what the blastula, this is the blastocells. So what actually happens in the invagination process? This part will depend. What? This part will depend to form like this. So what we can see is the same cells so we can see there is a formation of what new cavity. What? New cavity means a part of a cell is being infolded. What? Infolded over here to form the new cavity. The new cavity is being formed, and the new cavity is what communicating with the exterior. What? Exterior part. And this part is already being what the new cavity is lined by the already folded cells. And this type we can see same in the archenteron formation in the case of frog, the same type, that is the endoderm invaginations. Similarly, the sea urchin endoderm. So what are the things, what are the processes that occurs with the help of invagination? That is what the formations of the archenteron in the case of frog, whereas sea urchin endoderm formations in the takes place by invaginations. There is a second movement that is known as what? Involution. What? Involution. So what actually is involutions? So involution is the inward rotation of cell. What? The inward rotation of cells. So let me explain you how does it takes place. You have to remember all the examples that might be asked in the entrance exam. So let's say this is again about blastula figure. So what does it takes place? So the new cavity is communicating over here, that we have seen the archenteron formations. Then so like this is a new cavity. Now the new cell is being multiplying the blastoderm, it will goes like this. What? It will go in this directions and deposited inward. What? Deposited inward. Let me show you how does it takes place. So the new cells are being formed over here. What? New cells are being formed over here. Now these new cells what? Go into this direction. What? These directions. So what we can see, involution is the rolling of cell into interior. What? Interior part of the cell, that is known as what? The involution. And what are the process that takes place by the involutions? That is the famous is the mesoderm formation. What? Mesoderm formation in the case of amphibians. So amphibian mesoderm formation. What? Amphibian mesoderm formation takes place by involutions. Now coming on to the third process, that is what? The ingression. What? Ingression. So what actually is the ingressions? So ingression is simply what? The multiplication of the new cells, and the new cell is being what? The thrown upside into the blastocells. That is the migration of newly formed cell into the blastocell. So let me show you how does it takes place. So we can see this is the what? Typical blastula having a blastocells. So the new cells are being forming over here. What the new cells are forming. Now what is the fate of the new cells? The new cells will go into the blastula. What? Blastocell. Like this. So it will become like this. So like this it will go. So all the new cells that is go into the blastocell. What? The blastocell. And that process is known as what? Ingression. What? Ingressions. That is the migration of new cells into the blastocell. What? The blastocell. Blastocells. And what is the example? It is the same. The ingression is the sea urchin mesoderm. Whereas the mesoderm formation of amphibians takes place by the what? Involution process, whereas the mesoderm formation of sea urchin occurs by ingression process. Similarly, the Drosophila neuroblast migration. What? The Drosophila neuroblast migrations. So these are the two processes that takes place by ingressions. Now we're talking about the new process, that is the delamination. What? Delaminations. In case of Nepal, mainly we are being asked what type of this process, what actually is delaminations. We have given the definition of delaminations and we have to tick out the 4 options among the various processes. So what actually is delaminations? So in the we can see over here the blastula has a single layer of cells like this. Now it has to multiply. So similarly, let's say this is a fat man, with a big tummy. Now it has to lose the fat. Then similarly, what happens over here? This cell will undergo tangential division. What? Tangential divisions. That is what? The splitting of a single layer of cell into the 2 layers or the multiple layers. That is known as what? Delaminations. What? So delamination is the tangential division of cell into multiple or 2 layer. What? Multiple or 2 layer. What? 2 layer. And this occurs in the process of the famous process in the case of we mammals. So if you are, I wish all of you get admitted in the MBBS. So if you study the embryology of human beings, so you can see in the embryo there occurs what? Mammalian hyperblast formation. What? Hyperblast formation, through which there is what? Epiblast and hyperblast. So delaminations occurs in the case of mammalian and bird hyperblast formation. What? Mammalian and bird hyperblast formation. Now there is a last one process of the cell movements, that is what? Epiboly. What? Epiboly. What? Epiboly. So what is epiboly? So epiboly is a, so in the case of frog embryo or anything, we can see there are 2 poles. One animal poles having the micromeres, vegetal pole having the megameres. So it actually is the what? Covering up or the multiplying of cells or the inward rotations of the micromeres in order to invaginate or to the cover what? The deeper layer of cells. So let me show you the example. What? This is what? The micromeres. What? Micromeres. And this is the blue color plint is the megamere. What? Megamere has a larger cell, yolk clad in white in color. So in epiboly, what happens? All of the exterior part of the cell is being covered by the micromeres, and the cells which are in the bottom are being pushed into the what interiors. So this becomes what megameres is shifted over here, and the what this is the micromere covers. So epiboly means a covering of the or the rotation of the superficial layer of cells or the upper part of cells such that it covers the what the deeper part of the deeper or the lower part of the cell. So this all of the processes takes place in the place of phase of gastrulations. Now we are mainly being concerned with the word gastrulation of frog, that is being included in our HSCV course. So we're dealing about what gastrulation of frog. So gastrulation of frog is being occurring in the three phases. First of all is the emboli. Second one is the epiboly, and third one is the what contraction of blastopore. What the contraction of blastopore lip. So all these processes occur simultaneously. What simultaneously? Now before going on to these phases, we have to know about the one phase, that during the mid blastula phase, during the what middle part of the blastula, the cell divisions become slow and synchronous. What the cell division becomes slow and synchronous. Why this becomes synchronous? Because the cell has to migrate as a whole. That's why the cell will acquire which phase? G1 and the G2 phase. The cell will acquire G1 and the G2 phase. That's why the cell division becomes slow and synchronous. At the same time, there will be more formations of mRNA. What? The more formation of mRNA from the nucleus, which govern this process. It might be asked, which of the following phases of cell division is being acquired in the mixed blastula phase? That will govern to the gastrula. That is what G1 and the G2 phase, accompanied by more of the formations of mRNA from the nucleus. Now how this process takes place, I will be elaborating it with a figure. I will not be dealing with much of the text because you can get the text from the book. Let's say this is the figure of a embryo of frog, and the blastocele, this is the vegetal pole, and above this we can see this part is what? The neuroectoderm. What? The neural system, the fate map. This part is what? The notochord, and this part is the precaudal plate. What? The precaudal plate. Now this part, this whole part is what? The epidermis. What? Epidermis. The part that will be forming epidermis is not wholly. This is the part of the animal pole that will be forming what? The neural system. This part will form epidermis. This is the part of grey crescentia that will form notochord and the precaudal plate. Similarly, there is also again a part over here that is the somatolateral mesoderm. What? Lateral mesoderm. What? Lateral mesoderm. Now all this part is what? All this part is the presumpting endoderm. What? This part will form what? Endoderm. So I have taken a section from the dorsal part. What? From a posterior part. So let's say this is a blastocele. So I have cut out like this into the cavity, and I have rotated. What? Rotated. So I have taken a part from the posterior section so that all parts will be visible. Now what? How the process of gastrulation start? So for the process of gastrulations, or invagination or a part of the endoderm that is below the precaudal plate will go like this. What? Will what? Invaginate. What? A bit invaginate. That I have told you about cell migration movement. Now this invagin, at the point of invagination, there is a presence of two type of cells. Here is one type of cell, there is second type of cell, and there is what? A third type of cell. What? Third type of cells. So the type of cell which govern this process is known as what? Bottle cell. What? Bottle cell. This part is known as what? Superficial marginal endodermal cell. Superficial marginal endodermal cell. This part is known as what? Deeper marginal endodermal cell. Endodermal cell. So what is this bottle cell? So bottle cell is same like a Coca-Cola bottle. What? A Coca-Cola bottle. So what happens? The movement is towards the embryo proper. That is what is the body, whereas its neck is the part that will go in constrictions, and the mouth has what? A cytoplasmic strands that maintains contact with the outer side. Now this process is accompanied by what? At this process, what each process depends. So we can see a new cavity is forming or not. So I will draw this. You can copy the figure. Now what happens? This blastocele is being obliterating. What? This blastocele is being obliterating. Now a new cavity is being forming. What? A new cavity is forming due to the effect of bottle cell. At the same time, this mesoderm is also what? Involuting. What? Involuting is rolling of cell into the interior. At the same time, what happens? The outer part is being taken up by the neural sheath or the neural system, like this. The outer part is taken by what? The neural system. Now the notochord will be forming over here. This part is taken by notochord, and this part is taken by what? The precaudal plate. What? A precaudal plate. And the inner lining is due to what? The blastoderm. What? The blastoderm, that is the part of endoderm. What? Endoderm. So this cavity is known as what? Archaeonteron. What? Archaeonteron. Whereas this part is communicating is known as what? Blastopore. What? Blastopore. So what we can see, two processes are occurring. The invagination process means what? The deepening to form a new cavity, that is the archaeonteron. So archaeonteron formation takes place by invaginations, but at the same time the mesoderm is also rotating like this. So mesoderm formations is taking place due to involution process. What? So here occurs involutions, whereas the outside it is becoming what? Invagination. What? Invaginations. What? Invagination. So what we can see, the formation of two layers. Now again, what happens? At the same time, there is what? Epiboly formation takes place. What? Epiboly takes place. Epiboly means all of the superficial layer of cells will what? Contract or the try to cover the endoderm. What? Try to cover the endoderm. So the space is not increasing. What? The space is not increasing. That's why all of the outer part is taken up by what? The ectoderm. What? The ectoderm. At the same time, the endoderm will be displaced to the outside, or what? A bit outside. And this part is known as now what? The yolk plug. What? Yolk plug. So yolk plug is nothing but just a protruding part of the endoderm due to the migration of excess of the what? The ectodermal tissue. What? Ectodermal tissue, or the micromeres over the megamere. And this part is known as what? The endoderm. So archaeonteron has a lining of what? The endoderm. So this part is known as what? Dorsal lip of endoderm. What? The dorsal lip of endoderm. Dorsal lip. And this part will form what? Pharyngeal endoderm later on. And this archaeonteron will form what? What it will form? It will form the foregut. What? The foregut or the alimentary canal. At the same time, what this? The archaeonteron is being increasing. That's why the blastocele is obliterated. And at the same time, what? The mesoderm is also being rotating like this. What? Rotating. So at the last, what type of the embryo will shape it will give. So I will drop this. You can also see from the book. I am just clearing your confusions. So what happens? All of the outside is being taken up by what? Is being taken up by the animal pole. What? Animal pole. The inside part, the middle part is taken up by what? The mesoderm. And again the inside part is taken by what? The endoderm. Endoderm. And this is one is what? The yolk plug. What? Yolk plug. This is the archaeonteron cavity. What? The archaeonteron cavity. Archaeonteron cavity. So we can see like this. So this part is what the all of the part will form the ectoderm. This part will form mesoderm. This is what the all of the part is the endoderm. Internal part is what formed by the endoderm. This is the archenteron cavity. What archenteron cavity. So this process is all about the gastrulation process. If this video is helpful, so we can see the formation of three-germ layer over here. So if this video is helpful to you, then please like, and for more videos tap the subscribe buttons. I want to tell you all, I have got a lot of messages on my Facebook that my life is over and my name has not been placed in the merit list of IOM. I want to say you one thing: your life is just has been started. It has not been what stopped. You have just crossed what 18 or 19. Some persons have may crossed what 20. Your life has just been started. I have seen many of the students who have got their name in the merit list after 3 or 4 years. So do not underestimate yourself, do not lose your hope. Just what have a deep breath. You don't know how much stronger you are, how much potential do you have to cross any of the what the obstacles in life. And your aim is not only be to a doctor or anything. If you are not doctor, your life is not over, because life is much more beyond that. So let me just think of a world where everyone is a doctor. Then who will be the patient? Who will be the engineer? Who will be the writer? I know you are being passionate of wearing an apron and with a winding of a stethoscope around the neck. But in order to have this, you have to do hard work. You have to be very patient, because the seats are being limited and the emerging talents are being what are more talented than us. So you have to what keep on working on yourself. And if you have any queries about anything about the biology, mainly biology, then do comment. I will try to reply you as soon as possible. And stay tuned, connected with Akash Biology. Thank you.